I'm not recommending anything, I'm not making suggestions, anything, but I'm just opening up the floor. So feel free to knock me down actually here, right? But we need ideas and how to move forward from here, and how we want to open up, and how we want to leverage the scientists and the researchers working in the rice community to start using the tremendous amount of data sets that you've heard all over in the last 2.5, 3 days that people are generating for the rice, right? So I'm using some of the slides from the Rabbit Informatics portal, but this is have some of them have been modified for rice. And you heard something from Blake Myers on the first day, and you continue to hear all about the data sets that are being generated everywhere. So far you've seen that some of the repositories are like central repositories. You heard about Grameen, you heard about RAP, you heard about some other individual repositories. But as Ken mentioned in his slides, that you can replicate it at, like, for example, GigaDB, the genomes were deposited, plus they're being served out of Erie or Rise SNP projects, and then things like that. So the data can be replicated at multiple places so that users can access it. But another problem is that there is lots and lots of data sets and lots of small resources. Some are very dedicated and expertly curated. Some are not very expertly curated, but they have large amounts of data sets are also being there. So there is a distributed system of information all over the place. And I was just recording that over the last 2.5 days, I think we have heard about 20 or 25 different resources that people are hosting on their project or lab websites. So we think that the option was something like hierarchical federated data resources, means you manage it, but there are some hierarchies that GWAS data comes from certain places, and some of those collect information from their smaller projects as well, and then move it forward and allow the users to search for it. So this is something which Blake actually presented it, and you see that there is we think that the two gold standard genome data sets that people have often used is the IRGSP 1.0, that's going to be is the new one, and 9311 continues to be used, even though I think I don't know actually whether it is going to be resequenced or somebody has to redo the assembly of 9311. But those are the two major reference assemblies people are using, and more reference assemblies are going to be coming from at least 20 new genomes from the IOMAP project, right? So this is the one. Then you have stock centers that Ken mentioned about it. Then there are TRIM database from Taiwan, and then there are lots and lots of other mutant databases all around the world. There are about 6 or 7 or 10, about 10 rice stock centers, plus individual countries have their own stock centers. Then we heard from BGI a large amount of data sets. From GWAS, we just heard from Ken about the GWAS datasets, the Rise SNP project. We heard from Indian group today about some GWAS studies, and Masa presented yesterday about some of the GWAS. So there is lots and lots of datasets are coming. So those datasets are almost ready now. We heard lots of transcriptome datasets that are being reanalyzed and regenerated now. That is there. Interactomes, protein-protein or DNA-RNA interactions are there, molecular interactions are there. Obviously thousands of other genomes that are getting sequenced, but they may not be the referenced or finished quality. And then you start looking into epigenome, as you heard today, proteome and metabolomics studies picking up in the next 1 or 2 years. So these are the major data sets that you are having. Right now we have GWAS, which is pretty much ready. Gold standard genome data sets are ready. Stock centers are ready. Functional annotation of the genes and the genomes is happening as we read. Transcription data sets are, much of them are mature and are published. About I think about 700 to 800 or about 1,000 experiments and protein and molecular interaction studies. So I think for few of these things are already ready. So how we want to utilize these information for towards the annotation of the genomes, and how to annotate and help the plant breeders in some ways to move forward in their application in the field. So that's the goal of it. So far, so my timeline, what I think is that some of these are ready data sets. Some are going to be coming in next 1 to 2 years, and others would pick it up as we start building the homology modeling and things like that. So should we be hosting it at one place, which I don't think is a good idea. As Blake also mentioned, that it's not a good idea hosting it at one place. Nobody has the resources, nobody has the money to do it. But we need to depend on the partners. And the partners is whether we wait for the publication to happen or we release the data under some kind of an agreement that data is released as it is and you are not allowed to publish things like that. But to move the science forward, I think if my personal opinion is that next 4 to 5 to 10 years, peer review publication process is going to dissolve actually. The people and the user community will itself decide whether the data is a good quality or a bad quality, like a Facebook style like or not like kind of things. Peer reviews would be, nobody would have time to read the papers on those sites kind of things, because the data is so massive. And peer reviewers itself just read the papers, they don't reanalyze the data. So peer review process is actually failing at that point of time. So it's going to be changing. So, and some of the organizations who manage those things have to change as well. So this is how we are going to happen, is through the web services. You heard something from Ken today about using web services. You heard something from Saurav about using web services or semantic web and this is how the information is going to flow around most of the time, as I think. So I think the floor is open. How you want to approach the annotation and how you would want to build the resources. My suggestions are something like this. We have to move forward from genome and species level to the individual transcript and molecular level. So it's a whole gamut of from molecular gross level to the very fine level, base pair level annotations are there required. Query tools is the major option. There is plenty of data around. People go and browse, but they can't query the way they want to do it. So I think we need some very nice applications on how to query that data set to make some biological context out of that information, right? And then there are lots of resources who are building and annotating, but what kind of standards they are using in annotation, everybody has their own. So we need to come up with some guidelines on how you want to annotate and what needs to be annotated. Wiki approach that I'm mentioning here is one aspect, but you don't have to go forward about that, but there is something coming up. And who administers the data? I think individual resources administer their data and manage the quality. That's their responsibility. So that's the major thing. You can't be, most of the time I've heard is on the internet, everything what comes in may not be a good quality. So you take it with a grain of salt. So how do you trust that thing? That needs to be built in. And contributions from publication authors. We don't have the hands to curate. Saurav might have a couple of people in his team. RAP has 1 or 2 people to do the manual curation. Grameen has a couple of them. I don't know how many Kent's database people have on manual curation, probably nothing. Everybody who does it on the field, that is it. After that, the data is all handled based on the computational analysis. So there are not many hands, but everybody reads papers every day, right? So, and almost let's say in a month, about 4 or 5 rice papers, almost every rice researcher would read from their area of immediate concern, right? So at least they know the information and they're reading it, so they can curate that information plus anything that they are publishing, because they are the best users, they are the best people who know about that. So here's something which is going to change in a few about in a year or so. Is Grameen has a collaboration with the ASPB plant cell, and the idea is that author would be required to submit and annotate the data and the paper would probably not go up to the print until the data has been approved and accepted in by the major data repositories that host the data. If it's a GWAS data, we might send it to Oriza SNP or some other resource that who wants to manage it. Some genotype to phenotype data, somebody has to be recognized for that. If there is functional expression data, then they have to be managed and received by somebody we need to be recognizing, and they have to say, yes, we have received the data is in good quality and meets the standards that we would need. And then when you approve, only then the author gets a provisional acceptance of the manuscript. And that's going to be happening pretty soon, because that's the only time you can get the data out. Otherwise, they will just say, sorry, I don't have time to do it, or my graduate student has left, or postdoc is no more working. So that's not going to be an excuse anymore for the PIs. Then obviously there are lots of other, this is an Arabidopsis example, but we have reference genomes, we have reference gene annotations, but there are lots and lots of regions in the genome that are transcribed. They are known genes, or that your computational analysis of gene calling has missed them. So how do you annotate those things? There is no mechanism right now to add the genes, new genes that a user identifies on those things. So we need to come up with a strategy on adding those sections on those things. So I think this is a big hole about it. Nipan-Bari is I know that there are certain alleles in the Nipan-Bari which give you a truncated gene, or that allele gene is missing in Nipan-Bari. So if it's missing, how do you annotate that gene? There is no such reference for that purpose. So that's one annotation call to be done. And interaction, as I was saying, and I'll give you one example. This is from a human React on how the web services would work. So if you are, for example, a resource in Thailand or in Philippines is hosting a protein interaction database, for example, and they open up a web services, the plant reactome that Grameen probably is going to host, which we have been asked to, they can subscribe to your web services, and after that, once this is a React interaction regulatory network or set of genes is put together, we can say, here's my gene of interest, I want to see all the genes that are interacting with it. I don't have to go to another resource to go and query it. I can just say, open up here, select, give me a source like intact or bind or some other resource, like you have the Oriza friend. If they are serving the data, I can say, query that, show me all the genes that are interacting with this, and I will be able to find all those things up here in that sense. So that's one way of looking at it. So I'll stop here and ask you guys to tell us how you want to annotate and what you want to see put together for a RICE annotation project in that case, right? Suggestions, questions, open up. Yes. It's on.
One of the okay, yeah, okay. So one of the things that we realized while doing the curation is that the structuring of the data in publications is very heterogeneous in nature. Now, although we can develop a huge force of curators, at least that is possible in India, but we should also look forward to encourage the biologists to use a lot of ontologies so that we at least reach a level of organization as far as the experimental information is concerned, so that the curation process can be at least semi-automated in nature. Ontologies like plant ontologies, so we can encourage, we have to enrich the ontologies first actually, so that the experimenter can use it directly. Instead of saying leaf, he should just quote that plant ontology term, and that would make the process of curation very fast in nature. And the thing was that, as Pankaj mentioned, that journals, the collaboration from the journals are very important to get to close this gap between the database and the wet lab researcher. So, I mean, these are the few points that we have found during our exercise. and at least in India, we are trying to encourage uh, lot of people to come up and organize as curators, and we are trying to also train a lot of people, because this is something that is can be handled even as a part-time job. You need not come to the institute and work for it. You can work at a remote area. So that is from your own desk wherever you are working. Uh, for example, we have about 80 colleges in New Delhi who have a PhD in biological sciences, working as teachers. So those are the people which we like to target as becoming future curators, and so that is something that probably we'll be doing, and it would be good to have such exercises where we can increase the amount of curator community as such. So that was my point. Thank you.
Right. So I think the two suggestions came up as standardization of how the data is submitted to the journals as well as to other repositories, right? So data exchange formats. And the second one is how to mobilize the community to start annotating on their own, right? If I consider it second. So what we can propose is that as every journal allows you to submit supplementary files, so we need to come up with a strategy on standardizing the format of the supplementary files, so that you can just go and grab that file and run it through your scripts that you've written, so that your data can be imported into into your databases or any other archive that is interested in it. So that's one way of looking at it, and that's one of the goals with the ASPB. We want to work on it, and if you guys are interested, we would love to see on that side. And the second one, which I would show you how to mobilize the students and communities and authors. We can build an iPhone app actually. So as soon as you read a table of contents or you something know about the gene, you just click it on and you type it, whatever you say. Another option, which everybody's familiar with, Wikipedia, and which has been very, very successful. That community starts annotating the information, and if something is wrong, somebody else would come in and say, correct it, and you track.
A PDF viewer that was brought to our attention at at your plant ontology workshop in September. That's a very good example where publications from the Royal Chemistry Society have to adhere to the standard described by that system. So their tables and supplementary information are actually active tables. You can click on them, and if it's a graph, you can see the underlying data. It's right there available. And the links are active, and that viewer gives you the meta information on the publication within the viewer. Google Utopia. If you haven't seen this PDF viewer, it's quite nice. Maybe, Pankaj, do you have it there?
No, I don't have it here, but people should be able to look at the Utopia tool. It's a nice tool to work with. So, but I'm going to ask the Utopia guys to start including the ontologies that the plant biologists need in that source. Apart from that, and you also mentioned something about the ontologies, that there are not enough ontological terms to describe things. I would say then there are resources and feedback. Never ever be hesitant if you go to a website and if you find missing information or bad information. Never be afraid of writing back. That's the only way we know that you need something to be fixed. So there is a feedback or contact me. That's you need to do it right away to do it. And for ontologies that are not there, because we cannot. Whoever is developing ontologies does not know everything about that plant. So, and what kind of data sets are in the community. So you need to tell us, and there are trackers where you can submit the request for ontologies, and we make every effort to get back to you and involve you in the discussion on how to get that thing added to the vocabulary. So what they've been mentioning is that there is a fixed term of let's say leaf color Leaf color is a trade term. It has its own ID, and if I use the same term in any other database, I should be able to query the two or multiple resources using the same ID. But then I don't have to worry about whether you call it a, for example, I'll give you a good example of panicle color, panicle size. Then you have ear size, tassel size, then you have cob, and then you have other, all different cereal crops call their inflorescence by different names, but they're all inflorescence. So if I search by inflorescence, you wouldn't be able to get all of them, because panicle, tassel, ear, cob, whatever you call it, they're all synonyms of those of inflorescence in that term. So you would be able to query based on the synonym. So yes, you have a question.
What I feel is that the people who are working for almost their lifetime in a particular organism, they would like to refer their panicle, their inflorescence as a tassel and not as just an inflorescence. So that is something, that is where somewhere the ontologies also have to become flexible to get the feel of the biologist also. And just a reality check about how the data is available. Still papers in RICE are getting published which do not code any TIGR loci per se or a GenBank accession. They just mention the name. So that is something that we can very easily avoid doing that. But still publications are there which do not exactly pinpoint a particular LOC. We just name it by a conventional name. So I mean, at least we can get up to that level of organization.
Right. So on that side, Ken mentioned in his talk about the GCP application ontologies. Those are crop-specific ontologies. Let's say a rice community uses panicle color, panicle size, panicle abundance, and things like that. They are familiar, their breeders are familiar with those names. They don't want to see something different. So he also mentioned about reference ontology. So reference are the ones which would connect the two entities. You can call panicle color, you can call ear color, but how do you computer knows that these two are similar? That's the only way is to know is that inflorescence color is these two are instances of or synonyms of inflorescence color. So that's the only way to connect the two information. You would continue annotating using your respective terms, but that's the only way to query. The ID would be like that. So we are trying to build a flexible system on that side, and we would love to see more information about it. Yes.
So I personally, of course, I agree with your proposals, but I think the most problematic issue is this discussion is only from the bioinformatics sections. Actually, so we can provide many kinds of definition or terms on ontologies from our side. So we can use unified terms or definition, but users, not from our side, but experimental researchers. So without any support from their side, this kind of, how to say, development or this kind of discussion is still not enough, I think. Actually, when we see this room, actually at first many people are joining this session to hear everybody's presentation, but when panel discussion starts, many people go outside. This is the current situation. So we should not, I think we should notice that this is the current situation. So to better development or make infrastructure, not only for our side, but also from experimental researchers, we should discuss together, and we need to make this kind of situation or discussion time together, I think. So, yeah, and
make you feel better. I am a 90% biologist. I just moved into bioinformatics research. So we have a group who is working on totally biological aspects, but you're absolutely right that we need to get groups from biological sciences to as close to that.
Yeah. Thank you.
Is that a good question that I'm asking? Over here. Read it. I'm giving time to everybody.
And any comments from other persons?
Is that a good query that you would like to see? Show me all the genotypes with trait associated with it, that is salt tolerance, and the location, geographical location where they were evaluated. What kind of markers are associated with the traits and the genes associated with them, and how many are transmembrane genes located on the plasma membrane? Can you get me that answer from one single resource? And show me all the that's the big question that a breeder would like to ask. And breeder would also say, show me only in the genes only in this region of the chromosome, and show me only the ones which are dominant markers, right? And I want to see query the genotypes only by IRGC collection number, or the ones which are located only in India or in Thailand. Or even in Thailand, it's too big, so it'll say only in the ones which are in Chiang Rai region. So those are the kinds of queries people would ask in immediate terms in that case. So this is a kind of a very complex query. Can you do that thing? And the data would probably, the genotypes and the trait and the location and the markers may come from an ERISA SNP project or the project that Ken mentioned about it. Then the genes associated with that trait might come from Saurabh's database, and transmembrane location and plasma membrane can come from either Grameen or from RAP. So now we are querying 3 or 4 different databases to get you up to that answer, and I think that's a big question. And as a biologist, I would love to see this kind of a question. Jor, do you have any big question to ask? Something like that? How we can help you guys? So you need to come up with the questions for us.
This raises a very nice possibility. I was thinking about it, but you just summarized it. What a biologist would like to have. Okay, you asked this question from the breeder's perspective.
Yes.
But someone who is interested in deciphering gene function,
yeah,
then he would like to ask certain different questions, right? And all those questions, so rather than framing questions, the query model should be where possibilities are given. Suppose you mention a gene, then with this gene or with this trait or with this marker, these many possibilities are there in the database. And from where you want to form your query and which side you want to take your analysis. So query is there, data is shown, but analysis is not there.
Right.
Which I feel is lacking in all these databases. So combining all these databases should come to a level where a person can build analysis rather than querying. And query, sometimes you lose the way, but
right,
when possibilities are there, you know that with this particular trait or gene or marker or trait, these many possibilities exist in the databases. So wherever you want to take your analysis, you're free to take.
So what you're saying is that if I'm looking at this image, on something on very top are your 2 components, which I was describing your query. Another one would be your application, which does the analysis for you, because for doing the analysis you still need the data, right? So you collect the data and then you start putting it, saying these are the parameters that I would like to use for doing the analysis to build my hypothesis, or to validate my hypothesis up to that way. So that's what I was getting at. Thanks for bringing that question. It's a very important question because most of the databases actually allow you to search and query, right? They do not allow you to analyze the data. And every biologist is now struggling at this point of time. How do I analyze this data? This is so much of data for me to look at it. How do I analyze? And everybody's just building their own stuff. Some small applications, but not a big ones. But I agree. Somewhere in this, I should be able to put it another blob on the top. Software applications for data analysis. Galaxy is one. Any other? And somewhere in this middle, all these in the ideal world, all these arrows, there should not be any password protection. That's the very first thing. There is nothing my or mine. Everything belongs to everybody. Data is free flowing for me. That's the condition to get into this portal. You have something? So that is a concern, and that's why somewhere at the very bottom of this thing would be data archives, which still archive but not the actual data, the raw data, so that you can still redo the analysis in some ways. So any major data should be deposited or somewhere in the major archive. In that case, yeah. Yeah.
Yeah, sorry for interruption because of certain time. So, last comment, please. Yeah.
There was a meeting last month in Singapore in which it was a little bit of genetics, a little bit of downstream, so it was all about food security. It was about rice. And the concern was that, everybody knows that time is very short and we need a lot of improvement. So they brought all these Southeast Asia and South Asia countries together, and they had experts on crop modeling, on drought, and water mechanics and everything. So one of the common themes that came out was something that you're discussing, like the information portal for rice. And what they said is that this was organized by the Sloan Foundation in the National University of Singapore. And what they actually said is that they are really interested in funding something like that, but they would want downstream applications. So a breeder would be able to go in, somebody without a knowledge of bioinformatics, but be able to get the information that he wants from that. So I don't know if anybody knows about this, and I don't know how far it's gone, but the people who attended the meeting, they were asked to send in their ideas. So I'm not sure, but Dr. Ji Kang Lee was a part of that, and he mentioned this Sloan Foundation. So I just thought that I might mention it. And the other thing is, I'm from Bangladesh, from Dhaka University, and we are not a part of anything, but we are developing skills to do that. So we would be like to be a part in future. Thank you.
Yes, thanks for bringing up that information about Sloan Foundation. And I think we should keep track of it, and that has been brought up a couple of times in this meeting internally with Dr. Ji Kang Lee. So we are thinking about it, but if you are interested from your end in working and helping you out on this, developing some skills, I would love to talk to you about it. Yeah.
So thank you, Dr. Pankaj, jaiswal.